Hello everybody, welcome to the New South Wales Health Statewide Biobank seminar series. We'll just wait a few more minutes for people to join us and we'll start a little bit after 12. Thank you.
Georgia, I need you to either go outside or be quiet if I'm going to talk. Okay? Hope that was on mute.
That's all right.
It will certainly be edited out of the presentation for later viewing.
Yes, it's not easy at the moment. All right, everyone, look, it's 12 o'clock, but we will wait just a couple more minutes to make sure that as many people have joined us as possible before I introduce Andrew and he gives his seminar, which we're very much looking forward to. Look, I think we might start with our introduction. So again, welcome everybody to the New South Wales Health Statewide Biobank and our external seminar today. We're very much looking forward to hearing Andrew Gilbert, who is the Chief Executive of Bioplatforms Australia Limited, and I'm sure many of you already know Andrew, but I will just give you a brief introduction to his career. So Andrew is Chief Executive of Bioplatforms Australia, which oversees $300 million of Commonwealth Government research infrastructure investment in the discovery sciences across genomics, proteomics and metabolomics. And with 17 investments across Australia, Andrew has an extensive network of contacts from Commonwealth and state governments, along with prominent universities, medical research institutes, agricultural research institutes and also commercial entities. So the network supports 3,000 users every year, and this is across the spectrum of pure research to commercial production. So in addition to managing the National Infrastructure Network, Bioplatforms Australia has also catalysed the formation of a series of strategic national scientific collaborations, again in the broad themes of agriculture, biodiversity and biomedical science. Each Bioplatforms initiative is by design multidisciplinary, multi-institutional, and contains both discovery implications and pathways to end use. Innovation is inherent in both the model of collaboration and the environment that's created to foster excellent science and its application to important national challenges. So it's a great pleasure to welcome Andrew, who will be talking to us about the 3 R's, research infrastructure, research community building, and research impact. Thank you very much, Andrew.
Thank you, Jenny. And just to check, you can see those slides?
Yes.
Fabulous. Thanks so much. This is the first time I've ever had to give a seminar over video conference, believe it or not, a year and a half into these unusual times of ours. And I appreciate the invitation from Jenny. In fact, Jenny and I have worked around these types of research infrastructures and plannings for biobanks now for a few years together, and in fact sit on a committee or two relating to these things. So I really appreciate the opportunity. Some of you will know who Bioplatforms is, and some of you will not. We're an entity that was created way back in the middle of the 2000s to bring together a pretty disparate, sometimes competitive network of laboratories in the spaces of genomics, proteomics and metabolomics provision. If you haven't heard of us, you probably do know who the Ramashotti Centre is, or who the Australian Genome Research Facility might be, or you might have collaborated with a group like APAF at Macquarie University. In a sense, they're all us. We actually support a full range of laboratories around the country. And of course, respect that these sciences have become increasingly digitised, particularly over the last half a decade, and now we provide quite a significant bioinformatics infrastructure and network. As Jenny mentioned in her introduction of me, we also collaborate on and lead a number of very significant national research agendas, again, some of which you will be familiar with, even if you're not familiar with us. And I think we are also very committed to really a current mantra in Australian science and research support, and that's around translation and commercialisation. In fact, we're a tiny team. There's only 6 of us in the little Bioplatforms Australia entity, and that's our very fabulous team there. Top to bottom on the left is Sophie Mazard, Mabel Lum, myself, of course, Natalie Kurak, Sarah Nisbet and Sarah Richmond. And we effectively provide stewardship of a very significant Commonwealth investment through NCRIS, which I'll come to in a moment. We support really 17 physical facilities, although they're all reasonably autonomously run, and they reside through a variety of institutes around the country, and they hire in excess of 250 FTEs. I'll touch on later in this talk some of these framework initiatives, and you'll hear this terminology come back as I talk throughout the afternoon. But a very fabulous team. Again, you may have interacted with one or more of these people over the last number of years. So we were funded under the 2006 National Research Infrastructure Roadmap. Of course, I don't expect you to read this, but this is the 2016 roadmap. As you may be aware, there is a current government process being run to rewrite this roadmap as the 2021 version. It's done twice every decade. It identifies a small set of critical research infrastructures that are required to support national research demand and innovative opportunities. Bioplatforms is primarily supported through this complex biology chapter in this roadmap. We have very significant implications, though, in certain other dimensions of the roadmap, notably therapeutic development, some biodiversity studies through earth and environmental systems. I mentioned before we're increasingly digitised, and so we work very closely with these e-research platforms up here in the top left-hand side. To give you some more granularity, these are the elements in that roadmap that were specifically recommended for focus, in some instances ongoing support, such as us, and in other instances you'll notice that there's something here called networked biobanks in table 10, which was set up to explore opportunities through what was intended as a scoping exercise, and we might talk about that more in questions later on, but hasn't at this stage, notwithstanding very significant efforts by Jenny and others, hasn't yet fallen into a scoping exercise for future funding. But I think you should be very active and hopeful that biobanking can secure its place in the 2021 research infrastructure roadmap. I'm not going to read through this, and I don't expect you to. My slides will be available. NCRIS isn't about providing research support in the same way as, say, the NH and MRC or ARC or indeed MRFF do, but instead it's about providing the underpinning and enabling capabilities, both hard infrastructure, digital capability, and in fact, leading expertise that support the research system at large. Everything from discovery through to translation to post-production, quality control are all catered for within NCRIS capability. The awards are actually on a non-exclusive basis. So it's intended that these facilities become broadly accessible, and you would have seen earlier in my slides, we had about 3,000 discrete users last year and performed about 18,000 contracts. That gives you a sense that it's not one grant for one research project or one grant for one research team, or indeed one grant for one research institution. About 80% of our user base comes from organisations that do not own the infrastructure that's hosted in any given institution. So we've really taken these principles, particularly around non-exclusive access to HEART, and it really guides not what we do, but how we do it. And if you're interested in being involved in NCRIS in the future, I would highly recommend that you go and really brief yourself on your responsibilities. It is an opportunity, but it is a significant responsibility to the broader community in being involved in an NCRIS project. So the roadmap told us what we had to do, and the NCRIS principles tell us how we should go about doing that. Bioplatforms then went and created, well, we've been around for 14 or 15 years now, but we've evolved a strategy which has really got 5 specific dimensions to it. One is around laboratories. So I mentioned before genomics, proteomics and metabolomics. That's everything that entails, the full suite of DNA, RNA sequencing capability, from Sanger to short read to long read in its different flavours, as well as, of course, panels and so on and so forth. Likewise, in the mass spectrometry space, and amino acid analysis capability in proteomics and metabolomics. And in all of our laboratories, we have a mantra really of, first and foremost, we provide access. Secondly, we try and stay through innovation and adoption of global leading technology at the forefront of these fields. That's really our responsibility. And thirdly, it's one thing to own these, but it's another to make a significant strategic difference. So we're very focused on deployment, and I'll show you some examples of that in a moment. Science is always in flux, though, by definition, and these disciplines are in a very rapid flux. So we have a whole strategic element around identifying and adopting, and in some instances needing to raise funds to create new platforms. We were just very fortunately at the end of 2020 awarded 8.3 million to establish a national synthetic biology capability, effectively high throughput molecular biology, that focuses on metabolic engineering and new product development, materials or energy or whatever the case may be. This here says synthetic biology, but it might well be artificial intelligence and machine learning for the modern era of biology. So we have a constant scan on what next for the Australian research populace. Thirdly, I mentioned before that biology has become increasingly a digitised discipline. It's a bit of an irony because it's often said actually that people who are mathematically inclined tend to fall into things like astronomy or high energy physics, and biology is, for myself included, I think it's fair to say, biology might be a discipline that people who aren't so comfortable with high-end mathematics and physics and computational capability go into more observing science. But very quickly, in the past 10 years in particular, you can't do these types of biological analysis without deep digital skills. We've gone and created something called the Australian Biocommons, which actually puts in place the research infrastructures in behind the digitisation of the discipline. That's software and compute, significant data management and access capability for both human and non-human data resources, and of course significant training. And again, some of you might have been to our very extensive training effort over the years. The fourth element of our strategy is creation of data assets in support of really themes of some national significance. We don't just work in medicine, as you can tell here. We have responsibilities across environmental science and biodiversity, as well as food and agriculture. And I mentioned earlier, we have really taken to heart the need to translate these efforts into industrial settings. These national data initiatives do all of that. And finally, partnership development. We of course, are broadly accessible across the network for all comers, but we also focus very significantly on proactively joining into significant partnerships and granting opportunities. For instance, we're partners with the cardiovascular MRFF mission through the Australian Cardiovascular Alliance. We partner into CRCs, ARC Centres of Excellence, industry transformation hubs, and so on and so forth. In support of all of this, we've put in place things that you don't usually see in research settings, things like quality certifications. I would say now that 80% of our laboratory network has relevant ISO or NADA accreditations. Of course, we don't expect our metabolic laboratories at the Wine Research Institute to become accredited for human pathology. So they have relevant quality accreditations for their particular markets and collaborative opportunities. At a more specific level, we have, I'm not going to touch much on synthetic biology today, but these are the types of capabilities we have within each of what we call our platforms. I won't read through them one by one, but I'm pretty sure if you went to Canada or you went to the United Kingdom, in fact, if you go to New Zealand with Genomics Aotearoa, which is our peer over there, they have a very similar national network of capability that looks a bit like this here. And again, I'm not going to run through it one by one. These are the organisations that we support around the country. I won't read them out, but we don't, they operate reasonably autonomously from us, with the exception of the funding provision that comes via us and ANCRIS, but we're what we term as active investors. We work very closely with this network. We try and ensure that each of the platforms is integrated within one another, and in fact the disciplines work across one another in certain collaborative environments. It's very likely that through the pursuit of your own research that you've interacted and collaborated with one or more of these groups around the country. And when I talk about bioplatforms, I said before we're a very small team of 6 staff. We actually view this whole thing as bioplatforms. And we try and engender that each of these organisations, whilst they work for their host institute, view themselves as a participant in a true national program. We have collaborator or customer referral systems where groups that are at capacity or don't have the relevant skill base, actively and proactively refer work to other people around the country, other organisations around the country who are better placed to undertake that particular research enablement for customers and collaborators. This approach in biobanking, which by definition is broad and deep, will be very necessary, I think, and we again can probably talk about that later on if we get to some questions. So a bit of a snapshot of our system demand. We've now actually got 17 world-class facilities. Sorry about that. But we've got this national network of 17 odd labs. The Commonwealth provides about, for every dollar the Commonwealth provides, we attract about another $1.25 from host institutes and state governments. So it's very highly leveraged, and that's cash. We do consider in-kind as an important contribution in getting organisations to the table, but we don't actually count it much in our investment metrics. So we attract quite significant additional cash, a very broad-based activity around the country. In the case of our digital platforms, we now have about 10,000 users logging on to, for instance, Galaxy Australia, which is a software and computational platform, to use 10,000 discrete people logging on to use that computational platform every year. We've just completed our metrics for 2021, and we were either co-authored or acknowledged on just over 1,000 peer-reviewed publications for the year. So a very significant body of work is supported through the Bioplatforms network. I should also say we work very, very closely with our NCRIS peers. I showed you before in the 2016 roadmap that NCRIS is very broad in the capabilities that it provides to the National Innovation System. We work very closely, for instance, with the Animal Phenomics group. We work very closely with population health and therapeutic development organisations. In the biodiversity space, we work very closely with the Marine Observing System and the Terrestrial Ecosystem Research Network, and so on and so forth. And when COVID first hit last year, actually just about every NCRIS organisation, we didn't stop doing what we were doing before, but we got together and deliberately made an offer throughout all of the chief scientists, all of the health departments around the country, to the NHMRC, ARC, MRFF, of our support and contributions as requested into the national COVID response. And it was a very pointed example, I think, of the culture that NCRIS has instilled of enabling others first and foremost. And of course, in that particular instance, it was much required. At a specific level, last year we did a bit of a survey of the ARC Discovery grants awarded, for instance, and about just under 30% of those required omics in some shape or form. We often say there's about 30% of the Australian research workforce is focused on life science. So this, through a common sense optic, was about right. We partnered as a formal partner organisation on 3 of the 9 Centres of Excellence awarded in 2019. Just for transparency, they were peptide and protein innovations, native plant performance for agriculture, and synthetic biology. And of course, there's another Centre of Excellence round now, and I guess we've offered formal partnership. Well, actually, we do it on a non-exclusive basis, but we've offered partner organisation status to maybe half a dozen or 10 EOIs. So if you happen to be involved or leading a Centre of Excellence, and Bioplatforms is not supporting you, it's not deliberately, and please make contact. We partner extensively, as you would imagine, with CSIRO. What we do is required in about five of their 6 corporate priorities. MRFF missions the same. We're really delighted to be a partner on a recent Cardiovascular Frontiers award with Professor Gemma Figtree, who many of you might have interacted with over the years. We're partners on environmental science priority hubs. We work a lot with the industry growth centres and so on and so forth. I don't know how many of you are involved with the Zero Childhood Cancer program, but we are a strategic partner on that particular award that was through both the MRFF and Minderoo Foundation, where we provide all of the sequencing necessary for that particular large-scale or population-scale clinical trial for childhood cancer. And in fact, we are integrally involved in the data management, sharing, access and analysis, where we partner closely with the Children's Hospital of Philadelphia. So you can see a very broad base of work is supported, and really a specific set of examples there where we're deeply integrated within the research system. All right. I mentioned before that we create these framework initiatives. Some time ago, maybe 10 years ago, we decided that having such a significant network of laboratories and just renting time, if I could put it crudely, wasn't quite enough. And we needed to really deeply engage and deploy this national capability to science priorities of some national significance. And I'll give you a full case study around melanoma in a moment. But we do this across biomedicine, biodiversity and agriculture. This slide, probably you can't see these little icons very well, but you'll see when you look at them, these things are reasonably uniquely Australian. Most of you would have heard about the koala genome project, where we got the cover of Nature Genetics a few years ago under the leadership of Professor Rebecca Johnson, who was then head of science at the Australian Museum, and actually, wonderfully for her, is now the head of the Natural History Museum at Smithsonian. That particular study now is Australia's largest altmetric assessed project of all time. I forget the exact number now, but after 1 week it had 670 million views globally. So it gives you a sense that, and of course koalas are iconic, so it was a lovely thing for us to learn our craft around, but it gives you a sense of the interest in some of this uniquely Australian science. Of course, there's barrier reef here. We've extended koalas to all mammals in Australia, and in fact in time I've extended koalas to all mammals to now an ambition to sequence every vertebrate species for reference information, for phylogenomic purposes, and of course ultimately protection and conservation. In agriculture, we did a lot to begin with. We had a bit of a hiatus for one reason or another, but we've just commenced a whole lot of work recently on agricultural pathogens, on alternative crop types in alternative proteins and pulses, and we continue to engage closely with the agricultural communities around their priorities. And finally, medicine. I told you in a moment we would look at a more detailed case study in the melanoma space. We have worked quite significantly elsewhere though. Antibiotic resistance. We're long-term partners with the Australian Genomic Health Alliance that oversees the MRFF Future Genomics mission. We sequenced both the I'm not sure you probably are aware of the Asprey cohort led out of Monash. It's a healthy ageing study, very significantly and tightly phenotyped and biobanked. We sequenced the oldest, healthiest 2,000 people in that particular study and genotyped the full 18,000. We worked very closely and patiently with the National Centre for Indigenous Genomics for a variety of health and anthropological purposes. These particular deployments, they're valuable in their own right, but they're also valuable in building often very competitive research communities around a more singular view of how genomics and the other biomolecular sciences can help full communities of researchers make a more immediate impact, often by creating data in ways that conventional grants won't support. These collaborations persist well beyond our involvement with these things, and that's probably our greatest point of pride, in fact, is that we come in, we help do some good science, but we help broker really strong communities and collaborations, and it's a delight to us when we see them still writing grants and cooperating and publishing well some years after our involvement. Usually there's large-scale data resources that are left as persistent referential informations for communities of practice. Sometimes we store and host and make accessible that data. Sometimes that goes into relevant national and global data repositories. And finally, through our Biocommons investment, we develop and instantiate pipelines and workflows that are then available well beyond the collaborative purpose for which they were created, and they'll sit on things like NCI or PAWSI, the two supercomputers, or on Galaxy Australia, which is more a point and click workflow engine. I told you I'd give you a specific biomedical example of how these framework initiatives work. We were looking for something, I guess this was back in 2010, we were looking for something that was uniquely Australian, and I don't think I need to particularly describe why melanoma was chosen as a uniquely Australian challenge. But we came across a small but leading group of melanoma researchers, Graham Mann, who was then at the University of Sydney, Nick Haywood at QIMR, and some others, and got convinced that a contribution to the melanoma effort in the form of a referenced genome project would be highly valuable to support a raft of different research questions and opportunities. But we didn't want to support 3 people. So we asked Graham and Nick and others to bring together the national community, the best clinicians, the best researchers, patient advocacy groups, industry, and so on and so forth. And we had that whole community, it was termed a national lab meeting. We had that whole community describe out what we could in fact do that was a sentinel piece of work that would have value for all of them. And they kind of argued amongst each other behind closed doors before settling on something we were able to support. Bioplatforms contributed about $1.5 million, not in the form of a grant, but in the form of access to our network, where we paid the full cost of sequencing and data usage. Given the nature of melanoma, we were able to go away and raise an additional $3.5 million in 7 days, in fact, particularly through philanthropic sources. The Poach family was very generous. Cancer councils, of course, and once philanthropists and Cancer Council get on board, it was easy for the New South Wales state government to say yes also. I mentioned before we work closely with our NCRIS peers. We quickly collaborated with the NCI supercomputer in Canberra to actually take some variant calling pipelines that were then in place for the Chris Goodnow's mouse model effort at ANU and the Australian Phenomics Network, and they were able to tailor that for variant calling in human sources. We went ahead and sequenced about 500 pairs. There was a few trilogies in there as well, but about 500 pairs from the melanoma biobank, tumour and normal being a pet. And we created this data resource at that stage, and that data resource was processed and analysed here in Australia before being transferred to the International Cancer Genome Consortium data repository in the United States. At that point in time, that was the largest single contribution of mutational profiles into the ICGC data repository, and that data remains now open beneath the banner of ethics approvals and so on and so forth, of course, to all comers. Our learnings then, of course, didn't get lost within the melanoma community. We were able to transfer this to a whole variety of different programs that some of which I touched on earlier, the ZCC being one of those. The Garvan through the Sydney Genome Collaborative and generous support from the New South Wales government scaled out, did some more melanoma stuff, but also processed specimens from, I don't know, a dozen different projects through New South Wales. We partnered into the AGHA and have collaborated very closely with David Thomas over the MOST trial of rare adolescent cancers that he's had very significant research and clinical success through that trial, and I'm delighted to say we continue to collaborate with David Thomas and his Omico effort till today. In the case of our ANU lab, they actually were able to take what they'd done for our melanoma study and transition that capability into the ACT Health Genomics Initiative, and they're effectively the genomic pathology provider for ACT Health. So our investment became a very significant collaboration. It became a broadly national resource for a whole lot of purposes. The socio-economic benefit, though, is ultimately why you do any of this. And I'm delighted to say out of that particular melanoma project now, we've had 3 Nature papers, 3 very discrete papers, in fact. The 2017 one was around subtyping melanoma and its response to immunotherapy. And that's in fact changed clinical practice. I won't go into the details of it, go and find and read the paper, but it was able to describe at a molecular level why certain subtypes of patients will never respond to the current set of immunotherapies, and that changed clinical practice in its own right. I forget the statistics now, but those types of acral melanomas, they're called, fingertips and toes, account for about 4 to 5% of total melanoma patients, and it's in inverted commas, only a saving of 4 to 5 million to the health system per annum. But again, if you extrapolate out that 4 to 5%, as we work through the different subtypes of melanoma and indeed other types of disease states, you can quickly see that small changes in clinical practice extrapolate into very large dollar savings for the health system, and even better, significant health outcomes for patients. The second paper was a very big pan-cancer study undertaken through the national, through the ICGC, the International Cancer Genome Consortium that I was referring to before, where they looked at molecular dysfunction across a whole lot of different cancer types that until that stage had been assessed effectively through an optic of tissue type as opposed to mutational profile. That work will continue for a long time indeed, but that paper was not intended when we commenced our melanoma genome project, and it shows you the value of creating well-curated, managed and accessible data resources that permit science to be undertaken in the future that you hadn't envisaged when you got started. And in a way, this is why we call them framework initiatives. They're not projects, they're actually resources that permit a whole range of different activities to be undertaken over time. Finally, there was a Nature Comms paper, in fact, last year, led by Mark Shackleton. I think he's at La Trobe or Monash now, I can't quite recall, but he uses rapid autopsying, quite macabre, with help from Kay Confab down in Melbourne, and looking at metastatic melanoma disease progression under a hypothesis that we tend to try and treat these diseases with what we see, but we should be able to treat them if it's predictable on how the disease will predictably change in time. So we should be treating what is going to be rather than what has already happened, if I can put it that way. So that was a bit of a deeper dive into one of our particular framework initiatives that we're very proud of, and a nice example. We have nothing formal to do with this community anymore, but they continue to collaborate very strongly. They won, it's probably just ended now, NHMRC program grants, and so on and so forth post our involvement. And just finally, I know this talk had nothing to do with biobanking in particular, but we are, as a community, highly dependent, not exclusively, but highly dependent upon biobanking. That melanoma work was performed on the Australian Melanoma Biobank that's over in North Sydney there, next to the Mater Hospital at the Melanoma Institute. Our Exceptional Responders study that looks at certain patients in otherwise failed clinical trials that respond very well. It draws on highly curated biobank tissue from all around the country. It's particularly at the moment looking at response in lung cancer, and it's actually seeking certain types of responders to immunotherapy in lung cancer. So if you want to talk about that, please let me know. I mentioned before our work with NSIG. We need to access very historic Indigenous collections for that particular body of work. Biobanks aren't just biomedical though, of course. Our agricultural pathogen stuff depends on very historic, sometimes 100 years old and more, collections that reside deep in departments of primary industry and CSIRO. We seek germplasms and seeds from agricultural banks all around the world, in fact, and we're just partnering closely with DivSeek at the moment to do a bit of a landscape analysis of what material and what data exists where that we might be able to confidently draw on for future studies. I cross-referenced one of our facilities at the Wine Research Institute before. They work on yeast collection too, but of course grape and wine varieties, that they depend very heavily on very historic nurseries. For instance, the Yalumba nursery down in Adelaide, to draw on material for genotyping, flavour profiling, disease resistance, all of these types of things. And finally, I talked a little bit before about the koala work and our vertebrates initiatives. We also work on flowering plants and grasslands, and of course you can't just go and pluck a piece of wattle out of the bush. We really need to work closely with vouchered specimens out of botanic gardens and herbaria. And I think when ANCRIS is referring to biobanking, it's actually thinking about all of these things: the physical specimen, the digital description and management of these specimens, the quality accreditations that sits over the top of all of this, ensuring confidence in the science that's performed, and of course the accessibility regimes that make these things national resources rather than institutional projects. And with that, I say thank you so much for your attention. I know it's difficult over Zoom, but I welcome further questions and conversations. Thank you.
Thank you, Andrew. That was great. Now I'm wondering whether people have got any questions. Look, Andrew, I might start while listening to your talk. Can you hear me?
Yes.
Great. Listening to your talk sort of made me think about you know, the sort of requirements, the base requirements for national infrastructure. You know, we think about habitable planets. You've got to, I don't know, be a certain distance from the sun. You've got to have a certain kind of atmosphere. You got to, you know, etc. You know, from your experience, what are the kind of prerequisites for successful, stable, functional national infrastructure from that perspective, in terms of, you know, the research community that's going to rely upon that, but also the existing infrastructure capability that provides that sort of baseline to build something at a national scale?
Yep. There's probably layers to that, and layers of answer to that question, Jenny. That the first is that what type of science or medicine are you going to permit that wouldn't happen anyway? Well, why would government give you money for something that's happening? That's sort of a philosophical response, I know. The second is demonstrable demand and an extrapolation of that into future opportunity. Now, that future opportunity relates a bit to the first layer of the question, around what wouldn't happen unless this investment was soundly made. The third aspect is not, in fact, collections and freezers and descriptions, but culture, I think. You have to see your job in helping others, and there's huge rewards for doing that. In fact, I just last Friday had a bit of a social catch up in the afternoon with somebody who worked with me at Bioplatforms for a number of years, Catherine Shang. She worked at the Victorian Biobank. Now, she was a card-carrying scientist and now is a bit more like me, but when she worked at Bioplatforms, she got 3 Nature papers, no others actually, 2 melanoma ones and 1 around data management. So there's huge rewards in working with the very best people in the country. And Catherine, just by as an aside, went off to work with, after she left Bioplatforms, Oxford and Bayer on a translational program that they ran. So huge opportunities through embracing the right culture of openness, sharing and enablement. And then all above and beyond all of that, and it's the stuff that then starts to cost money. What in fact is it that you would spend that money on productively? And it most definitely is not on what your institute needs today. It's got to be, it can't be not that. It has to be that, but you have to think not what do I want, but in fact, what can I contribute through expenditure of funds? Lots of people will just want more freezers, but that doesn't make a biobank. Have to start thinking, how do I describe what's in the biobank in a way that makes it transparently accessible? How do I collect the metrics out the back end, because biobanks are finite resources. So if that's a finite resource that gets turned into tissue or DNA or data or publication, how do I catch that as a critical piece of data that relates back to that finite physical specimen as a legacy for that patient's contribution? So you've got to start thinking through the problem, I think, more than how do we collect and store another set of tissues.
Yes.
Sorry for the long winding response, but
no, I think that's very valuable. That's a valuable perspective. Toby, I can see you've got your hand up, so please, yes.
Yeah, thanks for that. Really interesting talk there, Andrew. And just actually perhaps building on that last point that you were touching on, that the expenditure, the funding for these institutions, which is largely should be directed at the outcomes that you're looking for, that what you're hoping to achieve with these outcomes. How just in an era where there are a lot of buzzwords that have to do with the economization of health and science industries and the like, these dictums like running business, running government, or running academia like a business. Are there ways of communicating this with, I would say, the larger institutions who are very risk averse where it comes to investing in these kinds of projects, or is it something that you're relying on so known quantities, people who have already shown their value in a research and a scientific capacity, are more capable of prosecuting that kind of argument for investment from larger institutions, and indeed turning that into the kinds of not just freezers but the roadmap forward, and having enough resources to actually achieve those outcomes?
Yes. If I can go to institutions first, there's no use being at an institution that doesn't want to do this. That's very simple. And most will have their biobanks, particularly the big ones, both medical research institutes and universities, for most of this audience. There's no use being there if they don't want to do it, so go find someone else. But where they do want to be there, I mentioned before we raise about $1.25 in cash. Often the Commonwealth money can condition how universities or medical research institutes would spend the money that they're already spending in a better way. More accessible way, and they get something in return for that. They get additional money, but sophistication, sustainability of platforms around what they're doing, and most big institutions now are starting to appreciate the value of research infrastructures and national research infrastructures, in fact, as persistent resources that help them do their research today and help them attract the best talent in the future too. So yes, you need champions internally. As for the translation or economisation of government investment, I actually think as communities of scientists, we get so used to being precise about everything that we've kind of got to cast that aside a little bit and start to be a little more entrepreneurial in our communications of value. And the economic value in doing stuff like biobanking isn't just efficiencies of scale. It's not just at the far other end of the spectrum, creation of new businesses and services and products, but I mentioned before savings against PBS on that melanoma study. We're able to, within a defensible spectrum, hand on heart quantify what that value is back to the health system. I mentioned before we're undertaking an exceptional responders project. That project is a bit about biology, but it's more about clever design of future clinical trials. And if we do that well, we're able to have less failed clinical trials, which reduce the future cost of drugs. It's telling this story. I couldn't quantify that for you today, but somebody could. It's telling this story and not worrying about the precision of it, I think. You still can't lie. You've still got to have evidence and confidence in the position you're prosecuting. But to think you need to have error bars that are within statistical acceptance is no Treasury official. You lost them anyway. They'll be lost. So I think it's not easy, Toby, but there's people around who know how to do this, who have done it before, that will happily help, I think, if you ask.
Great. Thanks very much.
Yeah, and I think that's a good point, Andrew. I think often preclinical researchers, we can lose sight of, the downstream cost of clinical care, which is infinitely greater. Preclinical research is very cheap. Clinical research is much more expensive. Clinical care, far more expensive again. And, those capacities for savings, in terms of, as I think you said, you don't need to really shift the needle by a great deal to potentially save substantial funds,
Yes, and there is groups. You've got your biobank there at Camperdown, but I noticed Wayne from Victoria Cancer Council, who actually Jenny and I sit on a committee of Wayne's, is here. They have really strong long-term metrics now over their user base, even by seeing the numbers of users out of industry. Industry doesn't spend money out of goodwill. They're doing it because there's a multiple of value in them doing that. It'd be pretty easy to extrapolate from that data as to the direct inflationary economic value that industry sees in accessing highly curated biobanks, I think.
Does anyone else online? Wayne, yes, please.
Well, I guess I just have to say thank you, Andrew, for mentioning us, and of course, thanks, Andrew, and both Jenny has been really helpful with the Victorian Cancer Biobank. I guess one of the challenge always for Biobank is really how to actually sell our value, and how do we actually work with everyone to provide that sort of as a research infrastructure rather than being, to sort of control that ends. I really find that having like case studies and so on, that's really helpful on selling that values with, and the way of how in the future is translating to that. Yeah, so thanks, Andrew. And so I guess the real interest is there with an economic evaluations is going to be good for a biobank to actually measure how's the output of that as well. Thanks, Andrew.
And it worked for Wayne. He's just secured his long-term funding through Victorian government.
That's right.
Yeah. Thank you.
Any other questions? Look, Andrew, I did want to sort of ask you about, just the general challenges of managing what will ultimately be in some respect a finite funding envelope in terms of balancing the support of existing national research infrastructure and acquiring new capability. I mean, that's something that when I've talked to people about, there's been, recognition of the fact that often the needs of existing infrastructure grows. We also need to support new infrastructure for the research of, 5-10 years in the future. How are those kinds of balancing decisions made?
If I knew.
With great difficulty, I guess, is the answer,
If I knew. How do I answer that? There's more money. I'm trying to get the figures at the moment from some contacts, but there's more money available between now and the end of the decade than there was for the last 5 years, and not just at an inflationary level. So there is scope within the budget futures, budget estimates to accommodate more. There is also, I've been dealing quite a bit with the roadmap exercise and the expert working group over the past few months. There is also a stated position of deliberately defunding stuff that's no longer a priority, internal to existing arrangements, as well as appropriate. And I don't know what is appropriate and what isn't, as appropriate shutting down whole existing, not shutting them down, but defunding them. So I think there is scope for new stuff, actually.
Right.
And for some disciplines, you can kind of say, well, our natural home is with the mouse models, or our natural home is with the astronomers or whatever, but there's not really a natural home for biobanking, I don't think, as it currently stands. So there's probably space for a well-crafted position to find a home, is what I think. But you need to do that as a collective. They're not going to fund 5 different things.
No.
Yes,
I think, look, we do have a few more minutes. I'll just see if anyone else has any last questions. Watching yes, Wayne.
Thanks. It's me again. I guess I have a question that I guess a lot of biobanks may always experience a bit of challenge of working with the commercial entities and the industrial, where because like what we mentioned is a lot of us, the biobanks are actually from the either research institution that find it hard that why would I want to give out the samples that we spend a lot of effort on actually procuring them and store them. So is there any sort of advice that's available for people who are actually keen to have some sort of collaboration with the commercial entity, especially from biobank perspective? How does that work with the experience from the NCRIST and so on?
Yes, it's important. It's important to respect people's motivations and drivers, but they do change over the time, particularly as they find value in an alternative approach. So an institution might collect a certain type of biobank or a certain subset of cancer tissues or whatever, but that institution inevitably also wants something from Jenny's statewide biobank in Sydney and the liver bank in Harry Perkins. And this quid pro quo is a very important driver. It's in fact called collaboration. It's a very important driver in the Australian research system. So respecting that people's drivers today are, as you describe, it's not hard to get on a trajectory where people actually find value, but find value in the collaborative network, but part of their response, and I use this word a lot, part of their responsibility in attracting and being custodian of some of the Commonwealth resource is opening up. I'm pretty positive those patients who generously and in some instances tragically donated those specimens wouldn't like it if they weren't available to the very best researchers trying to solve that problem elsewhere in the future.
I think that's a great place to end. Thank you very much, Andrew, for a really interesting talk. Thanks to everybody who joined us today. I've just put our link in the chat. That's where you'll find Andrew's talk after it's been processed by Toby. And also our future talks. I think our next talk is on Tuesday, the 28th of September, and we have an international visitor from Austria talking. Thanks again, Andrew. Thanks, everyone, and stay safe, and we'll see you soon, I hope. Thank you.